Drug design, challenges and approaches. Let's start by exploring an important trend in drug development. Over the last few decades, there's been a noticeable slowdown in the discovery of novel medicinal compounds. Why, you ask? Well, there are two main reasons. First, a strict empirical and rational approach to drug design. This means every step is precise and evidence-based, leaving no room for guesswork. Second, the high standards for safety and therapeutic efficacy, combined with the skyrocketing costs of research, development, and clinical trials. For example, biologics like monoclonal antibodies are highly targeted, but developing them can cost billions of dollars. Imagine the investment and effort behind creating something so precise. What is drug design? So, what exactly is drug design? Think of it as creating a tailor-made compound. The goal is to design drugs with a high chemotherapeutic index, meaning they are effective at low doses with minimal toxicity, specific actions that interact with particular biological targets. Instead of a trial and error approach, drug design focuses on a rational and logical process. It involves studying biodynamics to understand how a drug behaves in a biological system, exploring how drug molecules interact with biological molecules. Take statins, for instance. They are used to lower cholesterol. Scientists specifically designed them to inhibit the enzyme HMG-CoA reductase based on its role in cholesterol synthesis. That's precision at work. Objectives of drug design. Drug design isn't just about creating a molecule, it's about understanding how it works. Let's break it down. Molecular interactions and physicochemical properties. This explains how drugs interact at a structural level. Example, aspirin interacts with COX enzymes to inhibit prostaglandin formation, which reduces pain and inflammation. Processes. Producing pharmacological effects. This involves understanding how drugs achieve their therapeutic effects. Example, beta blockers reduce the heart's workload by blocking beta adrenergic receptors. Protoplasmic interactions. How drugs interact with cellular components to trigger a response. Example, insulin binds to its receptor to facilitate glucose uptake into cells. Metabolism and detoxification. This is all about how drugs are metabolized or eliminated by the body. Example, paracetamol is metabolized in the liver, but overuse can lead to toxic byproducts. Structure-activity relationship, SAR. SAR links biological activity to chemical structure. Example, penicillin's beta-lactam ring is critical for its antibacterial activity. Altering this structure reduces its effectiveness. The integrated approach of drug design. Drug design is not a single step, it's an intricate process. Let's look at the key steps. Chemical synthesis. Creating new molecules or modifying existing ones. Example, developing newer generations of cephalosporins to fight antibiotic resistance. Activity spectrum evaluation testing. How effective a drug is across various conditions. Example, broad spectrum antibiotics are tested for effectiveness against multiple bacterial strains. Toxicological studies. Determining how safe a drug is before it's tested in humans. Example, preclinical trials for anti-cancer drugs often include testing on animals for organ toxicity. Metabolism studies investigating how drugs break down in the body and the byproducts formed. Example, the prodrug clopidogrel is metabolized into its active form to prevent blood clots. Assay procedures, measuring drug concentration and efficacy. Example, ELISA tests are used to measure levels of monoclonal antibodies in the blood. Galenical formulation, developing the right dosage form for drug delivery. Example, long-acting injectable insulin formulations for better diabetes management. Broader implications of drug design. Drug design extends far beyond individual molecules. Here's what else it includes. Random evaluation in bioassay systems testing large libraries of compounds for activity. Example, high-throughput screening is widely used in the cancer drug discovery. Creation of new drug molecules using natural prototypes to develop new drugs. Example, paclitaxel, Taxol, derived from the Pacific yew tree, is a breakthrough cancer treatment. Synthesis of congeners, modifying drugs to enhance activity or reduce side effects. Example, second-generation antihistamines like loratadine cause less drowsiness. Isosterism and bioisosterism, substituting chemical groups to optimize activity. Example, adding fluorine to fluoroquinolones improves stability and potency. Precise design for receptor interactions. Designing drugs to bind effectively to specific receptors. Example, imatinib targets the BCR-ABL kinase in leukemia, offering a highly specific treatment. Challenges in modern drug design. But drug design isn't without its challenges. Let's explore a few. Cost. Drug development costs are skyrocketing, sometimes exceeding $2 billion per drug. Example, the cost of developing CAR T-cell therapies for cancer is incredibly high. Safety and efficacy. Regulatory agencies demand more safety data than ever before. Example, the thalidomide tragedy emphasized the importance of stringent safety protocols. Complex diseases. Diseases like cancer or Alzheimer's require multi-target approaches. Example, kinase inhibitors target multiple pathways to tackle complex cancers effectively. In conclusion, drug design is a meticulous multi-step process that combines biology, chemistry, and pharmacology. By focusing on rational approaches and leveraging tools like CR, bioassisterism, and high-throughput screening, scientists aim to create drugs that are not only safer but also more effective. Drug design is the art and science of saving lives, one molecule at a time.